These questions from the recent past papers. So these questions are recent past papers. Some questions are quite advanced and some questions are quite easy. So if the question's very easy, we will not much focus for easy question. But we try to answer the difficult questions. So how it work? Maybe you have forgotten still some of the information in unit 1. I try to focus the most important lesson in unit 1. Because if you are going to sit your exam, some of the areas are very important because questions are frequently asking from those areas. So how do you know frequently asking questions? This is teacher's experience. When teacher know, okay, this question has been asked several times. And difficult part is the questions related to practicals. I think many students cannot get good marks questions related to practical. Right. We try to do a questions here. I think you couldn't print this document. Maybe some might have printed, but some couldn't print. Doesn't matter. We'll organize in the future better way. So first, we are going to do this question 4. When you have a question, if you have a question or you don't understand something, you have a 2 ways to ask. One is you can directly ask by unmute your mic, you can ask. The second way is you can send via chat. I have this question, I don't understand this. By this way, you can interact to better understanding of the topic. Right, let's go. Now, this question number 4 is based on enzymes. Right now, we revise a little bit about enzymes and you can use this as your revision note. Revision note is a very short note which has very important thing. Right. Let's try to understand what is an enzyme. So here I'm going to draw a molecule. This molecule is a protein molecule. Enzymes are protein and you can see this protein molecule. And in this protein molecules, enzymes has a special place. The black color one is a special place. Right. Now you have to recognize this place. Now can you type and send what is the special place of the enzyme? There is a very special place of enzyme. You need to always remember this place. Here, the half circle represents the special place of enzyme. So now can you type and send it to me? What is this special place of enzyme? I got 3 answers. Answers sent by Senuka. Yes. Senuja Netuli. Right. Yes, your answers are correct. You should not forget this place. Active site. This is called active site. Now, why active site is important? Because this site is very important place for this round shaped circle. The round shaped circle one is called substrate. So the substrate, right? This is a substrate. Substrate. I think you can understand this in your chemistry knowledge. You will write a chemistry reaction A plus B, C. Simply, in your chemistry knowledge, you can write zinc plus oxide, zinc oxide. That's oxidation reaction. And you will refer these things in your chemistry knowledge reactant. And this one you call product. That's your basic chemistry. So in biology, we call substrate instead of reactant. Usually we don't call reactant, we call substrate. Now substrate and the active site nicely fit to each other. So these places very nicely fit to each other. So this substrate, which I'm going to write the substrate, is exactly fitting to the active site. This is exactly fitting to the active site. So after fitting to the active site, we can see it like this. This is the enzyme. Now this one is the substrate. Substrate is nicely fit into the active site now. After that, what is going to happen is not important for you, because in your exam, you will be never asked what's going to happen after that. What enzyme does to the substrate. This you're going to learn in your A levels. Here we are not going to learn, but outcome, the final outcome is we're going to learn what is happening. The final outcome is enzyme remain unchanged. So no change of the enzyme. Enzyme has no change, remain unchanged, but substrate become product. Substrate become product. So I just try to write in a different shape now. This is the product. Right. By doing that, what enzyme does? So what is the function of enzyme? What it does? So very important enzyme. This is a very important enzyme. Speed up reactions. So this is a function of enzyme. Enzyme speed up reaction. Okay, now what you have to do, you have to copy this note. After copying this note, you just send me a the chat message. Done. Done. That means you copy this note. So I know that you have enough time to copy this note. The enzyme, it has an active site, and substrate can fit into the active site. After that, they make this complex. So we write it complex. So enzyme substrate complex. Enzyme substrate complex. Then enzyme has no change and it make a product. So now substrate become product. That's very important. Substrate become product. Okay. You can copy this down. So I'm not going to revise everything of enzyme. The most important thing I'm going to revise that you need for your exam. Oh, just a minute. And I don't recommend some prefer to take a screenshot. I will copy this later. Don't do that. Do it at the same time because you will again forget to copy this down. And this is again learning. When you're writing in your book, it is a part of learning that you can remember after you draw this one and write in your notebook. You can remember easily. After few days, you can again read this one and you can remember many days. Okay, first one completed. Two completed. Yeah, okay. Many of them done. Yes. Yeah, I think most of them done. Right. I'm going to give very important hint or tip that is very easily that help you to understand questions and answer question. Before that, can you name one enzyme? Can you give me a name of one enzyme that you know? Let's see how many different enzymes I will get from you. Just to quickly type me a name of an enzyme. Senuco is first. Okay. Right. I got. Mhm. I got two names of enzyme. I got. Hmm, well, that's not enzyme, lithium. It's a hormone, right? We'll discuss later that one. That's a hormone. Yes, Senuja, it's an enzyme. Netuli, yes, it is enzyme. Senuka, yes, it is enzyme. Yes. Right. I think I got names of enzyme, some of the enzyme name. Amylase, yes. Lithium, amylase, amylase. Most of them amylase and one protease. Amylase and protease. Now, one hint is most of the enzyme end, the name end with ase. The most of the enzymes name end with the ase, A-S-E, A-S-E. So then when you see this end at the end, ase, then it is easy for you. This is enzyme, amylase, protease. So enzymes end with ase. This helps you to spot, this is enzyme. Okay, you can also copy this hint and hint enzyme end with name of the enzyme end with ase. So I don't have enough space to write the name of the enzyme. Name of enzyme mostly end up with ase. The word ase. So this is very important to identify. It is an enzyme. For example, I think someone say enzyme example is insulin. So I got one answer. Insulin as an enzyme, but insulin does not end with ase. So insulin is not an enzyme because it doesn't end up with ase. It's another enzyme. Peptidase, yes. Yes, peptidase also end up with ase. Peptidase, protease, amylase. Okay, so now we try to understand the question a little bit. Right here, pineapple juice contain protease. So you can see this end up with ase. So this is enzyme. So pineapple juice has enzyme, and this enzyme here they introduced to us as a bromelain. Protease. Protease mean they can break down proteins. So protease break down proteins. So it is important to know protease breaks proteins. Right. When you're learning digestive system, you have to remember table. So this table has all the enzymes: amylase, trypsin, pepsin, uh, amylase, trypsin, pepsin, and lipase, and maltase, and peptidase. Like many enzymes are there. So you have to remember all the enzymes. Okay, it's too much at the moment to remember everything, but you know, protease is enzyme which break down protein. A student uses this method to investigate the digestion of solid gelatin protein. Okay. So now it's easy for us to understand gelatin is a protein. Gelatin is a protein. Good. If you can eat gelatin as your dessert, you get a little bit more proteins because most of the gelatins, uh, they have a rich proteins. So now protein and enzyme is protease. Right now, student did this experiment. Now we try to understand this experiment step by step. Place solid gelatin protein into a test tube up to height of 5 centimeter, right? So I can take a test tube. I can dissolve gelatin in hot water. Then I can add this gelatin to 5 centimeter. And I can cool it down. Then as we make a dessert using the gelatin, it gets solid. Now after cooling, this make a solid mass, solid gelatin. So that's a height is 5 centimeter. Then I can also blend, can use a blender, pineapple juice, pineapple juice. Now something is important which you don't understand. Buffer. Now, how many of them have heard about the buffer? So just send me a yes or no, then I can explain you. So how many of them know what is buffer? No, no, not sure, not sure. Okay, I think most of them has no idea about the buffer. Right, we try to understand buffer like this. Before we come to a buffer, I'm going to introduce again two things under enzyme. factors affecting enzyme. So that's a very important topic, factors affecting enzyme. So what should you know about these factors affecting enzyme? I'm going to draw a graph. So this is my y-axis and this is my x-axis. And always you try to remember x-axis, we have to place our independent variable. Independent variable is what we changed. Don't forget, in experiment, what we change is independent variable. And here I'm going to always place dependent variable, what we measure. Right, that's a golden information. The dependent variable and independent variable. Believe me, still A-level student, they also mixed up this. Sometimes they use independent variable to y-axis, dependent variable to x-axis. Then the graph is wrong. You don't get the marks. So how many times I have to tell? I don't know, maybe 100 times I have to tell throughout this year until you sit for the exam, but still you make a mistake. You're going to mix up this independent and dependent variable. So independent variable, what we change in the experiment, dependent variable is what we measure. So I'm going to place here temperature. Temperature in Celsius degree. And I'm going to measure the rate of reaction. So we have a method. You don't want to know how I measured it at the moment. We think rate of reaction. I just place here rate of reaction. That is product form over the time. Product form over the time. So I'm placing 0 Celsius degree here, 100 Celsius degree here. And what will happen? The rate of reaction slowly increase and then go up to maximum. So then the maximum is rate of reaction is maximum here. So then I can check the temperature from x-axis. What is the temperature when the rate of reaction is maximum? So you draw a line to the x-axis and check the temperature. It is 37. You can read, this is 37. Then after that, you will notice it is decreasing. So the maximum level reached at the mountain, you can see the top of the mountain is the highest level. And you can see this temperature, 37. I think this one, many of them know. This temperature has a name. I think you know it. It's called optimum temperature. Right now, I know some of them know the explanation for this, and some of them know explanation from this. But half of the class usually they don't know. So why the rate of reaction is low at lower temperature? Why rate of reaction is again low at higher temperature? So temperature is high here. It could be a 70, 60 around here. It decreasing. And this could be around 10 degree. Rate of reaction is low. And you should know this because exam, this is a frequent question. You are always asking in your exam why rate of reaction is low in enzymes, why rate of reaction is high, rate of reaction is low in high temperature. Okay, we will try to answer this question later. Can you also copy this graph? Before we're going to learn about the buffer, you can also copy this one, the factors affecting enzyme. You can write a title, factors affecting enzyme activity. Number 1 is temperature. And then you draw this graph. After drawing the graph, we try to add some letters. For example, for this one is A, for this one is B. So we will later explain What is the reason for A? What is the reason for B? So why rate is low at cold? That is A. What is the answer for this? B. Why rate is low at high temperature? So there's a question left now. We need to answer this question. I know some might know the answer, but some they don't know the answer. But at the end of the day, at the end of the class, we all going to learn. So after you copy this graph, you can again send me, yes, yes, I did it. You can also write this question. We will try to find answer later. Why rate is low at cold temperature? Why rate is low at higher temperature? And once you copy this diagram, please send me a message, yes. And after 40 minutes, we have to leave from this meeting, and then again, please join again. So I use a free Zoom, so limit is 40 minutes. Okay. Litum senuka done. Okay. Litum senuka senuja nituli also done. Tamari, I think that name is your mother's name, maybe. Okay, I think almost done, all of you. And you can see here the logo here, Outstanding Pearson Learner Award. So IGCSE exam. If you get the highest mark, you have an award. For example, frequently students get this world prize for maths because some student they score 200 out of 200. So this is very frequent for maths. Even this time, I have seen few students score 200 out of 200 for maths because maths is very straightforward if you practice and you can always make your correct methods. Your answer is correct. You don't miss mark. But English, biology, chemistry, these subjects, physics, even like these subjects, it is very difficult to get because you have to really commit for it and you have to really understand for it to get the full marks, not 200, not 180, but it's good enough. Okay, keep in mind that as well, that you can commit for it to the something work. And in few seconds, we will be out again and we will come back again. To the last and second factor of enzyme is a pH. So pH, how pH change the enzyme activity is the second factor affecting enzyme activity. Then we can learn buffers. What is buffer? So